Today we are discussing about methods to detect potability of water samples. First one, introduction. Coliform groups. Coliforms, we are discussing about total coliforms and fecal coliforms. Total coliforms includes bacteria that are found in soil, in water and in human or animal waste materials. For example, Enterobacter, Klebsiella, Citrobacter, E. coli and Hafnia. Next one, fecal coliforms found in the gut and feces of warm-blooded animals. Example, E. coli, not found growing and reproducing in the environment. Next one, best indicator of fecal pollution than the total coliform, that is E. coli. Next one is streptococci. Fecal streptococci is found in stomachs and intestines of human and animals. Their presence indicate the presence of fecal pathogens in the water. Example, Enterococci. Next one, significance of testing coliforms. There are numerous waterborne pathogens. Individual pathogen numbers may be too low to detect in a reasonable sized water sample. Isolation and detection of some pathogens can take several days, weeks or months. Absence of one particular pathogen does not rule out the presence of another. Coliforms come from the same sources as pathogenic organisms. Coliforms are relatively easy to identify, are usually present in large number than more dangerous pathogens. As a result, the presence of coliform bacteria in water sample is an indication of presence of pathogenic bacteria. Next, standard qualitative procedures. Here we are observing prism 2 test, confirm test and complete test. First one is prism 2 test or MPN test, that is nothing but the most probable number or multiple tube fermentation test. In this, the most probable number method is used to check potability of water. That is, potability is nothing but the if water is safe enough to be drinking or not. The MPN method looks for the presence of potential pathogenic bacteria that may be in the water due to contamination of the water sample supply. Next one, MPN method enumerates the enteric bacteria called coliforms. Specifically, fecal coliform is nothing but the E. coli. Next one, MPN test includes 3 levels of testings. One is prism 2 test, confirm test and complete test. In prism 2 test, turbidity and gas in lactose broth was observed. In this, MPN or 100 ml of water using MPN table is present. Next one, confirm test. In this, growth on selective or differential media. We can observe the growth of the bacteria on the surface of the selective or differential medium. In this, uniquely highlights coliforms. Next one is complete test. In this, we are done gram staining techniques. Establish the presence of coliform bacteria. Next, coming to the flow chart, coliform test. First one is prism 2 test. That is nothing but the inoculation of lactose broth. In this, 2 types of results we are observing. One is gas producers, next one is no gas producers. In this gas produced, prism 2 evidence of coliforms examination continued, that is nothing but the positive test, gas formed. No gas coliform not present, examination stops here. Next, gas formed test tubes are taken and done confirm test. In this test, transfer made from lactose broth tube with gas to, here we are observing brilliant green lactose bile broth. This medium inhibits the growth of lactose fermenters other than coliforms. Thus, gas formation of BZLB medium constitutes a confirmed test that is coliform present. Next one, EMB, eosin methylene blue agar. Coli aerogenous organisms produce characteristic colonies, small, dark, almost dark centers with greenish metallic sheen. Next we will done complete test, that is the most typical colonies are select from EMB plate, then these colonies are placed in lactose broth and done gram staining. In this lactose broth, you are observing coliform producing gas or not. In gram staining, coliforms are gram negative non-sporulating bacteria. Then this test is completed. First one is Prism 2 test. Prism 2 test is nothing but the fecal coliform in the water sample by inoculating lactose broth containing Durhan's tubes with the water sample indicates the whether presence of gas or not. And here we are observing the gas producing microorganisms are mostly coliform bacteria. Here procedure, 3 steps of lactose broth are inoculated with varying dilutions of the samples. First step, 5 test tubes inoculated with, first of all, 50, we are taking 15 test tubes. All test tubes are inoculated with 10 ml lactose broth. Then among these 5 test tubes are taken, 5 test tubes are in the first step, 5 test tubes inoculated with 10 ml of sample, that is nothing but the water sample. Second set of 5 test tubes inoculated with 1 ml of water sample. Third set of 5 test tubes inoculated with 0.1 ml of water sample, and these 15 test tubes are incubated at 35 degrees centigrade for 24 to 48 hours. Flow chart of Prism 2 test. Here we are observing 15 types of test tubes. Here 5 test tubes are inoculated with double strength broth and 10 test tubes are inoculated with single strength broth. Here we are observing single and double strength. Single strength is nothing but the normal lactose bath. Percentage of the lactose bath is in normal condition. In double strength is nothing but the doubling the amount of lactose broth contained. Here we are observing 10 ml lactose broth in 15 test tubes, and then we are observing addition of 10 ml water sample in 5 double strength broth in 5 test tubes. Then 1 ml water sample in 5 test tubes and 0.1 ml water sample in another 5 test tubes. These 15 test tubes are inoculated with samples as well as placed Durhan's tubes. We are observing the slide material, test tube like slant materials is nothing but the Durhan's tubes. These materials are incubated 24 hours at 35 degrees centigrade. We observe the negative or positive result. We observing that negative Prism 2 test the absence of gas in both broth tubes indicates the coliforms are absent. Inoculation an additional 24 hours to be sure. After 24 hours of incubation, the tubes of lactose broth are examined for gas production. We are observing two different types of tubes. One is negative and positive. Negative Durhan's tube doesn't show any gap in that tube. We observe right side. Positive Durhan's tube, observe the half of the tube shows white color and half of the tube, bottom of the tube, shows the pink color. It indicates the presence of gas. Next, interpretation of the presumptive test results. Here we are interpret the presumptive test results. We can take MPN standard table, then only we can interpret the presumptive test result. Interpretation of the results. Positive test, those tubes that show turbidity with gas are marked as positive, and those with no turbidity or gas as negative. Positive marked tubes are counted from all three sets are nothing but the 15 tubes. One is 10 ml, next one is 1 ml, next one is 0.1 ml. The combination of positives in the three sets is used to find out the MPN or per 100 ml of water using the MPN table. For example, result is MPN index 553. 5 means that is 5 test tubes positive in 10 ml. Next 5 indicates, second 5 indicates the 5 test tubes positive in 1 ml. Next 3 indicates 3 test tubes positive in 0.1 ml. Here we are observing 553. That is nothing but the 5 test tubes. First 5 indicates the 5 test tubes positive in 10 ml. Second 5 indicates the 5 test tubes positive in 1 ml. Third 5 indicates the 5 test tubes, 3 test tubes positive in 0.1 ml. Here we are the predicted from the table, that is 920 colonies per 100 ml of water sample. Here we are observing the standard table. We are observing first one is 5 test tubes of 10 ml. Second one is 5 test tubes of 1 ml. Third one is 5 test tubes of 0.1 ml. Next, fourth column is MPN per 100 ml. Next, percentage of confidence. Here we are observing the result, that is 553. Now predict the result. In the left side, 5 of 10 ml, 5 we are taking bottom from 1 2 3. Third one from bottom, third one we are predicting 5. This is the 5 our result is 553. First one is 5, next one also 5, and third one is 3. The next MPN per 100 ml we can observe the 920 microorganisms per 100 ml. This is how the MPN result is predicted. Next one, sometimes the presumptive test also shows the false positive results. A positive presumptive test does not necessarily mean that membranes of the colon group are present. Members of the colon group are present. In most cases it is true, but there are exemptions. False positive presumptive tests are caused by the presence of other organisms capable of fermenting lactose with the production of acid and gas. Next one, bacterial association or synergism. Joint action of two organisms on a carbohydrate result in the production of gas that is not formed by the either organisms when grown separately, gram positive or negative. Next one, elimination of false positive presumptive test, that is nothing but the addition of triphenyl methane dye in the lactose broth medium inhibits the growth of gram positive bacteria. Next one is conformed test, done to determine the origin of coliforms and they are E. coli or not. Media: brilliant green lactose bile broth, EMB agar, or endose agar. Procedure: eosin methylene blue agar prepared by adding 2 dyes eosin and methylene blue to melted lactose agar. Then a loopful of the culture from the lactose broth with gas is streaked over the surface of EMB agar plates. The plates are incubated at 37 degrees for 24 or 48 hours. Coliforms produce characteristic colonies on EMB agar. E. coli produces small colonies, dark, almost black center with greenish metallic sheen. Enterobacter produces large pinkish colonies, dark center, rarely shows metallic sheen. Now we are observing 2 different types of microbial colonies. Left side is on EMB agar. We are observing bluish green metallic sheen is nothing but the E. coli. Next one is pink mucoid colonies. We are observing Enterobacter. Positive test is nothing but the appearance of least one typical colony, or if no typical colonies are present, at least 2 atypical colonies. Next one, complete test. The purpose of the complete test is to determine if the colonies appearing on EMB agar plate are again capable of fermenting lactose with the production of acid and gas. If the organism transferred to an agar slant shows the morphologically property of coliforms. Procedure: the typical or atypical colonies from EMB plate are inoculated on lactose broth and agar slants and incubated at 37 degrees for 24 to 48 hours. Next, interpretation of results. Here we are observing formation of gas in Durhan's tubes, positive test. Next, nutrient agar slant, gram staining, gram negative non-spore forming bacilli leads to the positive test. Result is confirms presence of coliforms. Flowchart: here observing the flowchart, confirm test and complete test. Here no gas produced coliform groups are absent. Positive test: gas production, use positive confirm tubes to be determined MPN. These positive tubes, all positive presumptive test cultures used to inoculate tubes of brilliant agar lactose broth incubated for 48 to 48 hours at 35 degrees centigrade. These positive tubes are inoculated on plates of EMB agar and incubated at 35 degrees centigrade for 24 hours. Then observe the presence of colonies. Then these colonies are inoculated in agar slants as well as Durhan's tubes. Okay. This is the total flow chart: presumptive test, confirm test and complete test. Here we are observing the presumptive test, that is 15 test tubes inoculated with inoculum and Durhan's tubes. Then here confirm test, we are observing the positive resulted from presumptive test are inoculated in EMB agar and observe the colonies. Next, complete test, these colonies are inoculated on slants and broth tubes. Then observe what is the result present in that particular water. Advantages and disadvantages. Advantages: correlated to the presence of pathogens. Coliform population is large enough to isolate in small water samples, for example 100 ml. This is the rapid test. This is the inexpensive test. This is the safety not culturing pathogens. Next one, disadvantages. Labor intensive, large amount of glassware is required. It lack of precision, large errors, still require survival and culture of organisms in lab. Next technique is nothing but the membrane filter technique. Here we are observing this technique involves filtering 100 ml of drinking water through a special sterile filter. In this we are observing special sterile filters are observing. These filters are made up of nitrocellulose acetate or polycarbonate are 150 micron thick and have 0.5 microns diameter. When the water sample is filtered, bacteria larger than 0.45 micrometers diameter in the sample are trapped on the surface of the filter. That it indicates that the viruses are not trapped on the surface of the filter. The filter is then carefully removed, placed in a sterile Petri plate on a pad saturated with a liquid or agar-based medium, incubated for 20 to 22 hours at 35 degrees centigrade. It is assumed that each bacterium trapped on the filter will then grow into a separate colony. By counting the colonies, one can directly determine the number of bacteria in the water sample that was filtered. Here we are observing the pictures membrane filter technique. Left side is test sample, is filtered membrane, microbial traps, different types of microbes, and filtrate is observed in the bottom. In right side, that is, we are observing a special instrument that is consisting of sample to filtered funnel-like structure. Then the bottom of the funnel you are observing the membrane filter retain cells. Next one is conical flask with vacuum. Then after that the filter is removed and then placed on the surface of the particular medium and incubated. Interpretation of results. Use the proper medium allows the rapid detection of total coliform, fecal coliforms or fecal streptococci by the presence of their characteristic colonies. A grid pattern is typically printed on these filter discs in order to facilitate colony counting. This grid-like structure is used for the easy counting of the colonies. Here we are observing three different types of Petri plates. Then A1 coliform reaction on an endomedium. B1 fecal coliform growth on a bile salt medium containing aniline blue dye. Next C1 fecal enterococci growing on an azide containing medium with TTC, an artificial electron acceptor added to allow better detection of colonies. Advantages and disadvantages of membrane filter technique. Here we are observing the advantages: good reproducibility, single step results often possible, filters can be transferred between different media, large volumes can be processed to increase assay sensitivity, time saving are considerable, ability to complete filtration on site, lower total cost in comparison with MPN procedure, Disadvantages we observing: high turbidity water limit volume sampled, high population of background bacteria caused overgrowth, metals and phenols can absorb to filter and inhibit growth.